We have Dr. Timothy Rogers from Department of Psychology of University of Wisconsin-Madison. He's going to talk about connecting natural and artificial neural networks in functional brain imaging. Thank you for joining us today, Dr. Rogers. You're ready for your talk.
Thank you. It's my pleasure. I really appreciate the invitation to talk to your group in the symposium. In particular, I admire your foresight in planning an online symposium before it was cool, before we were all locked in our basements doing science. I'm especially interested in this, talking about this particular topic. It's a relatively recent branch of research that draws on some issues in cognitive science, neuroscience, and also machine learning. And so I'm very keen to get feedback on what I'm talking about today, as this is all new work that's in progress. And it's also work that when I was a graduate student learning about neural networks in the 1990s, I thought would never happen. I didn't used to think it would really be possible to connect natural and artificial brain networks and functional brain imaging. But lots of people are doing it now, and I'm excited about it. So I thought I'd start with my mental picture of functional brain imaging today. Here it is. It's like the Wild West in America, in my perspective. So the Wild West was a very exciting place to be. There was a new frontier with lots of new discoveries to be made, but also maybe a somewhat dangerous place and lawless with a kind of lack of order. And I think all those things are true of functional brain imaging today. Why is it exciting? Sure, I don't need to tell you, but it seems like, you know, every other month there's a high-profile paper coming out from a group that's combining machine learning and the various ways that we have of measuring neurophysiological responses in the brain to try and decode how neural systems encode different kinds of information. I think there's a real sense that over the last 10 years or so, some important conceptual barriers have been breached, and there's a lot of textbooks that are going to be rewritten as a consequence. Why dangerous? Well, at least from my perspective, one of the issues is that a lot of sort of contradictory and difficult results are coming out of some complex workflows across different labs. I'm just going to use some of my own work to highlight, not to go through this image or model, but this is a paper that we published a couple years ago, combining neural network modeling and functional brain imaging to try and flesh out some components of the cortical semantic network. Anyone who's familiar with my work probably knows that we've made a big deal of the anterior temporal cortex here as being very important for understanding how semantic information is processed in the brain. And right around the same time, there was a high-profile paper that came out of Jack Gallant's lab, applying a sort of natural language processing, Bayesian decoding, and brain imaging techniques in a very sort of sophisticated analysis that yielded these beautiful images here, with the idea that the semantic network kind of tiles the entire cortex in some way. And just looking back and forth between these two things, you can see that the two perspectives look somewhat different, which is not in itself a problem. That's kind of science, right? But when you start to inspect diagrams like this one, you can see up here in the upper left, there's a kind of color code for interpreting the kind of information that different patches of cortex are meant to be encoding. And there are certain kinds of results that just seem puzzling from the perspective of cognitive neuroscience. So if you look here, green is meant to indicate visual information. It seems to be encoded here in the dorsolateral prefrontal cortex, according to this map. If you look back in the occipital lobe, where most of science tells us visual information might be represented, you can see this kind of purplish color, which according to the code is meant to indicate representation of mental state information, which seems unusual. And if you look at my favorite area of the brain, the anterior temporal cortex, you can see this grayed out color basically means it's almost the only part of the entire cortex that doesn't really have a lot of semantic information in it. And so across these two studies published within a year of one another, they're both using relatively contemporary techniques that are combining machine learning and brain imaging, but they're leading to really fundamentally different views of how semantic information might be encoded in the brain. So that's exciting, but it's also worrying, right? If every lab has its own set of results and its own workflow, how do we reconcile what these different kinds of patterns mean? What we need is like a new sheriff in town, right? I say that facetiously, of course, but what we really need is some sort of causal modeling framework that will allow us to understand how and why different workflows in different labs achieve different kinds of pictures of how information is represented in the brain. And so when I was thinking about these issues a few years ago, I was excited because I think some of the new techniques might allow us to make use of a long tradition of research using artificial neural network models in cognition and neuroscience. Incidentally, I can't see anyone, but if there are questions or if you're having trouble hearing me or seeing anything, don't hesitate to raise hand or just let me know verbally. I'm also happy to take clarification questions at any time. Okay, so these are just a random sampling of neural network models that have been influential in cognitive neuroscience over the years. I won't go through them all, but what I want to suggest today is that these kinds of models where we have really quite a good understanding of how they work and how they might be used to simulate human behavior might help us to bring some order to cognitive neuroscience, functional brain imaging, and the analysis of other kinds of neural signals. So there are two pieces that I'd like to tell you about today. One has to do with functional magnetic resonance imaging. I'm going to talk a little bit about why analyzing fMRI signals might be challenging from a neural network perspective. I'm going to show you some simulations comparing different kinds of contemporary approaches to decoding neural signals. To assess whether they meet the challenge, I'm going to show that they have some important blind spots. And then I'll introduce a new approach that we developed here in Madison that yields some, I think, provocative results when they're applied to real brain data. And then depending on time, the second part concerns applying a similar approach to understanding neural dynamics that occur in ECOG signals. These are electrophysiological signals recorded directly from human cortex. So let me just start with the first one. I'm going to show you a super simple model, which is a kind of almost cartoonishly reduced version of this kind of model that I've used for many years to understand semantic cognition. So this is a model that learns bidirectional mappings between visual representations of objects and verbal descriptions of objects. It's a fully continuous and recurrent deep neural network. You imagine that when you see an image, it activates some features. The activity propagates bidirectionally throughout the network and gradually activates units that locally represent individual words like this is a dog, it's furry, it's friendly, it has a tail, and so on. And because the connections are bidirectional, you can also provide as input verbal descriptions like a dog, furry, cute pet. Activity will propagate throughout the network and it can generate visual representations of the item described, like so. So I said cartoonishly simplified. What I'd like to do is show you this cartoonishly simplified model and use it to exemplify a a series of challenges for functional brain imaging that I think arise if we really believe that artificial neural networks are useful scientific models of how brains might represent and process information. So here's my cartoonishly simple model. We imagine, let's say, visual images are distributed patterns over some bank of units that propagate their activity to some other bank of units that encode, let's say, verbal propositions about objects via some hidden layer. And in this case, I'm going to imagine that the hidden layer just has 2 units so that we can plot their activity patterns graphically. And let's imagine that we train this simple cartoonish model on 4 objects like a boat, a car, a bird, and a dog. And because there's some semantic structure in the patterns, the inputs and the outputs for the boat and the car should be somewhat more similar to one another than they are to the bird and the dog. And the bird and the dog should be more similar to one another than they are to the boat and the car. So you can imagine training this model. And now treating it like a little mini brain that you want to do functional brain image analysis on. And I'll just show you some of the internal representations that might arise for these 4 different objects across the 2 hidden units for different runs of the model, where the only thing that differs between runs is the initial pattern of random weights sampled to start the model off. So on the first run, you might see that unit number 1 is showing this pattern of response to the boat, car, bird, and dog. So roughly similar, but little differences between boat and car and bird and dog. But unit 2 is really apparently seeming to encode the distinction between the living and the non-living things. So unit 2 might look like a little living thing detector. But if you reinitialize the model and run it again, you might get a very different solution. So here unit 2 looks like it's hardly doing anything. Unit 1 looks like it might be behaving as a living thing detector. In run 3, you might get something like this, where each unit looks like one's detecting non-living things, one's detecting living things, and so on. And so if you imagine that each run is kind of like a different subject in a functional brain imaging study, and you do what people have done in functional brain imaging for many years, which is you align the units. Let's treat each unit as being similar to a voxel in a brain imaging study. And we can measure the activity of each unit for each pattern. And now what we want to do is find units where the mean activation is higher for, let's say, living things compared to non-living things, or vice versa. Well, if each network is like a model of a single subject, and we simply average the activity for, let's say, unit 1 for boat and car versus bird and dog, if you run this several times, you get nothing for the hidden units, right? Because the particular way that any given unit responds to these different items is essentially arbitrary, then the mean activity for each category can end up being essentially the same across many different network runs. But that sort of averaging at the unit level is obscuring the important fact that I'm sure you're all aware of, that in every run, the distributed pattern across both units is always expressing the same similarity structure amongst the training patterns. So what I'm showing down here at the bottom is just the same activity patterns here, but instead of plotting them separately and independently for each unit in this simple model, I am plotting them geometrically. So activation of unit 1 and unit 2 visualized as a 2-dimensional space. When you look at the data this way, you can see that every model is always achieving the same internal representation, and all that's varying across runs is basically the orientation of those representations in the model's internal representation space. So in other words, the common technique of looking for single individual voxels or areas that exhibit mean differences in activity pattern could be obscuring important signal if what's really happening in brains is that representations are distributed over many ensembles of neurons. Many others have pointed this problem out, of course, but I'm just going to list these two things as challenges for functional brain imaging. One is that if brains are like artificial neural networks, then information can be coded over representational ensembles, that is, in patterns of activities over many different voxels, whose individual elements may not be independently interpretable. And secondly, it may be the case that even immediately adjacent voxels are not encoding the same information in the same way. It may be that one voxel is coming on for animals and its neighbor is going off for animals, or its neighbor is not really distinguishing animate from inanimate, but is distinguishing birds from dogs, let's say. And finally, the particular way that a given voxel or a given region might encode information across individuals may be different, even if the ensemble, the patterns, are always capturing the same similarity structure. So these are three issues that have been apparent for a long time, but they're not the only ones. Here's a cartoon of a neural network as you might see it in a textbook or a paper, where units are grouped nicely into layers. The layers determine the graph structure, the kind of the different units. So all the green units here are receiving and sending connections to the same banks of units. And it's really the connectivity that determines which units get to cooperate together to represent and process information, right? We think of these green units as being a layer because they get inputs from the same subset and they send outputs to the same subset. But one of the things that's interesting about networks is that the same connectivity can exist in a graph even when the topographic or spatial layout of the units is very different. So in other words, if I just move these units around like this, as long as the connectivity is the same, the function of the network will be exactly the same. In other words, the green units in this configuration should still be treated as jointly encoding some representation because they get the same inputs and they send the same outputs, even though they might be topographically separated in space. And likewise, an organization like this, where the green and yellow guys happen to be interleaved, just because they're spatially interleaved, it's not their location that matters, it's really their pattern of connectivity. So again, the green units should all be treated as jointly encoding some information. In functional brain imaging, we often use spatial proximity as a kind of kernel for smoothing data or for averaging or reducing complexity of the data. If real natural neural networks have this kind of property that components of a representation can be separated in space or interleaved with other representations, then that's smoothing, blurring, or averaging is going to be highly problematic. And of course, we know that brains are very kind of densely interconnected with white matter pathways that allow neural populations that are really spatially segregated to rapidly influence one another, raising the possibility that representations can really be topographically separated. So the fourth challenge for functional brain imaging, if neural networks are good models, is that representational ensembles, groups of neural populations that jointly encode some information, might be non-local, anatomically dispersed. And as if that weren't already enough, it could well be that the particular spatial or topographic layout of a given network varies across different individuals. Of course, we all share the same genes and the same sort of gross blueprint for connectivity, but brains are also plastic. And so the particular fine-grained way in which different little neural populations come to represent or encode information, although we would expect them to be grossly similar at a coarse scale, at a more fine-grained scale, we might expect some degree of individual variability in which units take responsibility for which bits of their input and so on. And so this is really a problem for any functional brain imaging approach that is averaging signals across individuals at a given location. It's assuming possibly wrongly, that the same information is encoded in the same location and in the same way across individuals. So the spatial layout may vary across individuals. And just one last thing is something that we all know about, but in papers that illustrate neural network models, of course, these models are only containing units or features that are relevant to the computation that we're interested in. When it comes to real brains, the whole problem is that those neural populations, units, what have you, of the network that's actually involved in the computation that we're trying to study, are going to be buried amongst other neurons, other neural systems, other neural populations that are doing something else. And so there's a kind of needle in a haystack problem where we can't really look at patterns of activity across an ensemble until we happen to know which of the measurements we've taken should be treated together as a group that are behaving as an ensemble. So the networks of interest constitute only really a small proportion of the measurements taken. So my picture of functional brain imaging back when I was learning about neural networks and thinking about these issues for the first time was that, representations, information is distributed over representational ensembles. So you can't look at them one neural population or voxel at a time. You need to look at whole patterns. But those patterns could potentially be anatomically dispersed and intermingled with other ensembles. And they might be somewhat variable in exactly where and how they get situated across individuals. And they're buried in vast amounts of irrelevant data. So for a long time, I really thought that if neural networks were good models of neural systems, then functional brain imaging was really facing a difficult problem. So sad clown here. Of course, the enterprise proved not to be hopeless in many different ways, right? Certainly there are lots of robust findings, replicable findings in the literature. So just empirically, there are results that we can collect and replicate. But on the other hand, there's also a huge amount of variability and lots of failure to replicate that we see. And so what we need to do is to understand what that means, right? What does it mean when different kinds of statistical methods, machine learning or decoding methods applied to the same data set deliver different results? And this is where I think neural network models can help. So what I want to do is show you a simple model that we trained up, and then we did functional brain imaging on it to kind of evaluate what kinds of signal different sorts of methods can discover. So the model is just an autoencoder model, right? So you put some pattern across the input, activity propagates forward, it has to just copy the pattern to the output. And what's interesting about this model, simple though it may be, is that we construct input and output patterns that belong to two different categories. You can think of them as being any kind of categorical distinction that you might want to seek in brain imaging data. Let's say half the images are faces and half the images are places. So in the model, there are two different ways that distinction between these categories, A and B, can be represented in neural activity. Okay. One is, in the input and the output units, we have some bank of units where the probability of being active if you're in category A is marginally higher than the probability of being active if you're in category B. So these are really independent localist codes. The mean activity tends to be higher for A items relative to B items. And right next door, we have some input and output features that show the reverse. They're a little bit more active for B than A. And then we have a bunch of input and output units that are just random vectors, no structure at all. Now the arrows indicate the connectivity of this feed-forward model. So there's one pathway that's really only connecting these input and output features that independently encode the category structure. And so the hidden units in this pathway are going to learn a distributed code in which the A items tend to be, on average, more similar to one another than they are to the B items, but with no guarantee that any single unit in this hidden layer is going to show more activity for A items than B items. In other words, this is a distributed code where viewing it as a multidimensional space, the A and B items will always be separated. But unlike the input and output units, there's no guarantee that single localist units will be more active for one item than the other. In this other pathway, it's only connecting the arbitrary units here, and it also gets to talk to these output units here so that if there are any kind of weird variation amongst different items in the outputs, the arbitrariness of this representation can help to encode individual differences between category members, which allows these units to be sort of even more directly expressive of the category structure. And finally, we imagine that this network, which is computing the mapping from input to output, is coexisting with a bunch of other units that are uninvolved in the task and just take low random values. Okay, so this is our little mini brain. And if you've understood what I've said so far, the question is, if we use this network to generate simulated neural signals, so we just look at the activity of each unit, take it as the bold response at a voxel, distort it with a little bit of Gaussian noise, then we can do functional brain imaging on the network. We give it a bunch of items from category A, a bunch of items from category B. We simulate the bold response at each unit for all of those items. And now we apply a method to try and figure out which units are encoding the distinction between category A and category B. That gives us a way of assessing how well different brain imaging methods are doing at finding these independent localist codes like the input and output units versus finding distributed codes like these hidden units. And what's more, we can look at the success of those methods in this case where all of the interesting information is nicely localized. So we imagine that the hidden units are all anatomical neighbors of one another, as are the input and the output units. I'm going to call this the localized layout. And we can also look at their success in finding that signal in this more interesting case that I characterized earlier, where the green units that are encoding distributed information about the category are anatomically dispersed and are intermingled with other kinds of units that are encoding other kinds of structure. Okay, so I'll just emphasize in both of these cases, the network connectivity is exactly the same. Okay, all that we're doing is changing the assumed spatial or topographic anatomical location of where these units reside in the brain. Okay, so in these simulations, as you can see, super simple feed-forward autoencoder model. There are 35 items in the A category, 35 in the B category. We train the model to a low error criterion. We train it 10 times with different random initial weights, and we treat each trained model now as a different subject in a brain imaging study. Okay, so we imagine we have 10 subjects. The only thing that's different is the initial starting weights, and as a consequence, they can learn internal representations that vary. And now for each model subject, we record the unit activations generated by each of the 70 items at every unit. We distort with Gaussian noise, and then we analyze the data to find the quote unquote voxels or units that are encoding the distinction between A and B categories. And the question is, what methods will find all of the different signal carrying units? So here's univariate contrast. This is just looking for units that are on average more active for A than B or vice versa. I'll just skip these details. In this plot, here's the ground truth, just to remind you, right? We've got red units are more active for A, blue units are more active for B, green units code A and B, but in a distributed way. Golden units are just arbitrary input-output patterns, and gray units are inactive patterns. So here's the result we get for univariate contrast. And the way I plot these data, any colored circle was deemed to reliably encode category information by the method. If it's red or blue, that tells us that the method also uncovers the direction of the code. So red tells us that A units were more active on average than B units. Blue shows us the reverse, and green will depict units that the method detects, but it can't tell what direction the code is. And then I'm showing this for the case where the hidden units are localized on the left, and the case where they're dispersed anatomically on the right. I'm just plotting them in exactly the same way, not like this kind of mix up thing so you can see what the results are. So with that orientation, you can see the univariate approach, of course, not surprising to anyone, is easily finding most of the input and the output units. This bar plot is showing the hit rate minus the false alarm rate, right? So how many true positives did it get and how many correct rejection minus the number of false alarms. So 1 would be perfect discrimination here. You can see it's doing quite well for the input and the output units. But of course, I hope this isn't a surprise to you. This univariate contrast really is completely missing the distributed representation in both of these cases. Okay, so this is not new or surprising. This is the whole motivation for using multivariate pattern classification to decode neural information. So let's look at some common multivariate decoding techniques on these same data. So the first one, this is now getting more interesting, I hope. The first one is the Searchlight pattern classification technique that Nico Kriegscourt, Francisco Pereira, Ken Norman, Matt Botvinnik, and many others have made such great use of in recent years. The method, if you're not familiar with it, works as follows. The idea is for each voxel, you take all other voxels that are within some anatomical radius, okay? So we're looking within a kind of sphere. You take all of those voxels and their activations across the different items, and now you just fit a pattern classifier to look at the pattern evoked by each item and to try and decide whether the item belongs to category A or category B. You fit the classifier, now you test it on some held out set, and you store the holdout accuracy at the center of the searchlight. And you now compute that holdout accuracy at each voxel within a single subject for all of the neighbors of that voxel. You do this for each one. So for each subject, what you get at every voxel is a number that tells you how well can I classify A from B given my anatomical neighbors. And you do this for every individual subject. And now what you're looking for are individual voxels that reliably have a classification holdout accuracy that is above chance across subjects, right? So in other words, you average the classification accuracy across subjects at each voxel location, you get a confidence limit on that accuracy, and if the accuracy is above chance, you say, okay, in general across subjects, this neighborhood can be used to decode what I'm looking for. So if we compare this to the univariate contrast, we no longer are assuming that neighboring voxels code information in the same way, right? You can have any pattern across those neighbors. As long as those patterns are capturing the right similarity structure, you can decode. Likewise, it doesn't assume that you can interpret the activity of a single voxel independent of everything else. You are looking at ensembles, right? Whole patterns across many voxels at once to try and decode them. But on the other hand, you're still assuming that the information has to be localized in the same way across subjects. But the information has to be contained within at least one of the spheres that you're looking at, because you're looking at different searchlights independent of one another. And so it also assumes that the information will be anatomically clustered. It's not clear that the method will be able to pick up things that are anatomically dispersed. So this is the result I showed you before with univariate. What happens if we apply the Searchlight pattern classification approach to these simulated data? What you can see is that in the localized case, again, every colored circle is a unit that the approach determined coded reliable information. Here they're coded green because you can't tell what direction the code is, right? All you can tell is, can I decode A from B given my neighbors or not? But with that caveat, you can see that the model, this is with a Searchlight size of 7 units, which is exactly the size of this hidden unit. It does great at finding the distributed representation, and it does pretty good at finding the input and output units. But if you make those distributed representations anatomically dispersed, so they're no longer locally clustered, now it's right back down to chance again. I can't find those guys at all. And furthermore, the particular result that you get really does depend on the size of the Searchlight that you're using. So here's some results from a smaller Searchlight and a larger Searchlight. If we make the Searchlight smaller, it continues to do well finding the distributed signal, but it's now starting to fail at finding the input and output units. This is because the category coding is quite weak and the data are noisy. If we make the Searchlight larger, it does great at finding the input and output units, but it completely mislocalizes the signal from the hidden units here. Basically, the Searchlight is so big that Searchlights that are centered on these irrelevant units capture all of these guys. And in this particular simulation, that's the only place where the signal got identified, even though the signal that was being exploited was in these other units. So there's some advantages to Searchlight, but it's still going to have problems, especially finding anatomically dispersed signal and being a little bit susceptible to an arbitrary parameter choice the size of the Searchlight. So this brought us to consider, why are we using Searchlights at all? Why don't we just take all the voxels in the brain, take the measured response for each item that we care about, and train a single classifier to discriminate A from B. And now we can use model regularization as a technique for making sure that there's a unique solution and that the optimization is convex and all the rest of that stuff. So if you're not familiar with this kind of approach, the idea is, rather than pre-selecting a small group of voxels and then fitting a classifier, we take all the voxels at once and we fit in this case, we're using regularized logistic regression. So what does that mean? You're trying to find for every voxel some coefficient beta such that the product of the data times the coefficients gives you, passed through a sigmoidal squashing function, gives you a number bounded between 0 and 1 that minimizes the discrepancy between the true category label, which is binary, 1 or 0, and the predicted label, which is the sigmoid function of the data times the beta coefficients. Right? This is just normal logistic regression. In decoding brain states, the problem, of course, is that you have way more features than you have data points. And so you need to, so in other words, there's basically an infinite number of possible beta coefficients that will perfectly reduce the error. So what you need to do is constrain your model fitting by some other cost that tells you which amongst that infinite number of solutions is most likely to be a real solution. So we looked at a bunch of different vanilla kinds of regularizers, but the one I want to tell you about right now is the L1 or Lasso Regularization. So the idea is you're trying to find coefficients that reduce the error, that's this term, right? So good prediction, but also jointly minimize an additional cost, which is just the sum over all the coefficients of their absolute value, okay? You want to make that sum small. How can you make the sum of a bunch of absolute values small? Well, you can make a lot of them 0, right? So one way of minimizing the L1 norm is to put 0 coefficients on as many voxels as you can. And so optimizing this cost is essentially doing model fitting and a kind of feature selection at the same time. Any voxel that has a non-zero coefficient has to be useful in reducing error. Any voxel that has a 0 coefficient is not necessary for classifying the items. That's the logic. There's one free parameter in this model, which is this lambda, which is a constant free parameter that determines how much you care about the solution being sparse versus reducing error. And we tune that parameter using a nested cross-validation procedure. So if you do this on the model, I'll just mention the assumptions of this approach. Doesn't assume any kind of localization. It doesn't assume any kind of consistency across subjects or anything. The only assumption it makes is that the signal is overall sparse. There's a small number of features or voxels that matter for the problem that you care about. And so fitting a model with just sparsity gives this result with the current simulations, which is not great, right? You can see, so again, colored units are units that the method identifies as being important across the 10 different subjects. In this case, importance is determined through permutation testing. And you can see like the things it identifies are real signal carriers, right? It finds 2 of the distributed units, 1 of the output units. But there's a whole bunch of stuff it fails to find. The hit rate is very low. The false alarm rate is 0, but the hit rate is very low for both kinds of things. And why is that? That's because the L1 regularizer only cares about sparsity, right? Because there's quite a lot of redundancy in the signal. There are many voxels that are all coding roughly the same thing. The L1 regularized decoder can show quite good decoding accuracy with only a small handful of all the important features. So decoding with L1 gives an overly sparse solution. So this is the point we're at when we started talking to our colleagues in optimization signal processing in the engineering department. So this next approach is one that we invented with Nikhil Rao and Rob Novak from the engineering department in our university. Chris Cox was a grad student working with me currently at Louisiana State University. And we call it the sparse overlapping sets lasso. It's related to multitask learning and the overlapping groups lasso, if you know those approaches. The intuition is that we can fit a whole brain model with a regularization cost that is better informed by what we expect the signal to really be like. What do we expect the signal to really be like? Well, we think that the signal carrying voxels, we don't expect them to be in exactly the same location in every single person, but we do expect them to be in roughly similar locations across subjects by virtue of all having the same genetic plan for our brains. So in other words, if I know that there's a voxel in this part of my, well, sorry, you can't see me pointing, I'm sure, but in a certain part of my brain, that makes it more likely that neighboring voxels in my brain and neighboring voxels in your brain might be signal carriers. They don't have to be in exactly the same spot, but roughly the same spot. So we want to design an optimization that prefers solutions where this is true. We can do the model fitting not for single subjects one at a time, but for all subjects at once. We warp their spaces to a common brain volume, and now we fit a single classifier to all voxels at once in one giant whole brain optimization. We fit a single classification model to all data, but we use a regularization penalty that's going to encourage some degree of cross-subject similarity in where the voxels are located. How does that work? Well, I'm going to give you the intuition, and then I'll briefly show you the math. The idea is, if you imagine these are different, each row is a different subject, the colored squares are signal-carrying voxels. The idea is that the optimization is going to prefer to put a non-zero coefficient on a voxel if it belongs to a group that also has a lot of non-zero voxels. Groups are defined just the same way as searchlights, right? They're just other voxels that are within some fixed radius of a given voxel. So this square here is denoting a group containing these voxels for all different subjects in the same space. And these groups are used in the definition of the regularizer such that placing a non-zero coefficient on a group that is densely occupied like this incurs a low cost, but placing a non-zero coefficient, let's say on this voxel, which belongs to a group that is otherwise unoccupied, that's going to incur a high regularization cost. Okay? So I'm just going to go back for a second. You might have the question, well, how do you divvy up the brain into groups? What's elegant about this approach, in my view, is that the groups can overlap. That is, each voxel can belong to multiple groups, and each group contains multiple voxels, right? If you look here, here are two groups. These three voxels belong both to group 1 and to group 2. When you're computing the regularization costs, you compute it over each group separately. And coefficients that get attracted to one of these features will end up being counted in the most densely occupied group. So let me show you what that cost looks like. Again, this is regularized logistic regression. We're minimizing this function where there's some free parameter times a regularization cost h. But here's the definition of that regularization cost in the SOS lasso. It's a function of the coefficients and their grouping into sets. That's S here. The total regularization cost is a sum over sets, and that's just one of those groups. Now within each set, what we're doing is we're basically taking the elastic net loss, okay? So we're taking the sum of the L1 norm. So this is the same as L1 regularization here, just the sum of the absolute values of the coefficients. This is going to pressure as many features to 0 as possible. But we're also now taking into account the L2 norm as a loss. So this is the sum of the squared values of the coefficient. The thing about L2 is that it likes to make coefficients small, but it never pressures them to 0. So in other words, if you have two features within a set that are perfectly correlated, rather than putting all the weight on just one of them and a 0 coefficient on the other, the L2 norm is going to want to put equal weights on both of them so that they're both small, so the sum of their squares is small. So in other words, this term wants to spread coefficients across the group. This term wants to pressure as many coefficients as possible to 0. Those two terms together are weighted by another free parameter alpha here. So it's just a mixing parameter for the two costs. We tune it through nested cross validation. That cost is computed within each set separately for every set. That gives us the whole regularization cost, and then we find a set of beta coefficients that minimize that total cost. Okay, so if you're not familiar with those kinds of approaches, the basic idea is that the thing is liking solutions where the features tend to be anatomically grouped, but it will also tolerate solutions. If the only way to reduce error is to have them very dispersed, it will tolerate those kinds of solutions as well. And we can see that in the simulated data here. This is the solution we get for the SOS lasso. And you can see it's not getting absolutely everything, but it's really getting most of the features, signal carrying features in the left branch of the network here, with very few false positives, both in the case where the signal is anatomically localized, but importantly, even in the case where the signal is anatomically dispersed, it's finding that distributed signal almost as well, in fact, just as well as when the signal is spatially localized. So what does this tell us? The key point is this. I've made the case that neural networks suggest that representations may be distributed in a way that is invisible to most of the current statistical approaches we use to decode fMRI data. Univariate contrast, Searchlight multivariate pattern classification, and regularized whole brain multivariate pattern classification. I showed you the L1 norm, the L2 norm, and elastic nets have other problems that I haven't gone into. Since neural networks suggest that signal can be distributed in this way, and we know that you can't find that kind of signal with these methods, that raises the possibility that functional brain imaging in general may have only given us a limited picture of how and where information is encoded in the brain. It's not to say that any of those results are wrong, but they may well be incomplete if neural networks are a good model of neural systems. And finally, a consideration of the ways that neural networks might represent information suggests a new approach for decoding neural signal using these structured sparse models. The SOS Lasso is one example of this class of models. There are other approaches that one could take. So the sort of million-dollar question in this project is, if we apply this new SOS Lasso approach within some domain where information is thought to be highly localized, we can now assess whether in that domain the representations are really more distributed than have been previously recognized. So that's the next piece that I want to tell you about. We looked, as so many people have done, in one of the domains where very strong arguments for functional localization have been made by Nancy Kanwisher and her group. I'm talking, of course, about the fusiform face area and the associated face-specific systems that Kanwisher has proposed exist as sort of modular representations of faces in posterior temporal and occipital cortices. This finding is so well known, it's even on Wikipedia. This is what you get if you look at face representation in the brain. Here's a quote from the Nature Review's neuroscience paper in 2008, which is a nice summary of this view. We propose a model in which overlapping continuous maps of simple features give rise to discrete modules that are selective for complex stimuli. What I want to ask is whether this is a valid model, given that functional brain imaging, which has provided a lot of the data for this perspective, may have been unable to detect more distributed signal. So in the current study, we borrowed some data from Brad Postal's group. This is a paper from Jared Lewis Peacock and Brad Postal, where they showed people pictures of faces, places, and objects, asked them how much do you like these things by button press. So the response is not related to the category. And in particular, what I like about this data set is they used a slow event related design. So there's something like 30 seconds or so between each stimulus. They could measure the entire peak and decay of the bold response generated by each stimulus. So we have no deconvolution or statistical modeling beyond just choosing the bold response from the fifth TR after the stimulus presentation. So it's raw bold with minimal processing. And we took that bold response from all cortical voxels. What I want to show you is, for the exact same data set, where does the information appear to be when you look at each of the different statistical approaches that I outlined with the model? So I'm showing here at the top, these are the face system in red and the perihippocampal place system in blue, maps that we downloaded from Nancy Kanwisher's site. Kudos to her for publishing these maps and making them available. So this gives you a sense of where the signal should be under this localizationist view. Here's the result we get when this is with the Lewis-Peacock data set, when we apply the univariate contrast approach. The blue here is the result we get from a whole brain false discovery corrected solution, with blue showing areas that are less active for faces than other things, and they line up pretty nicely with the perihippocampal place system. And then over here, what we get is a reliable ROI analysis when we just look at the fusiform face area here as reliably more active for faces than other stimuli in this data set. So this looks like a replication of at least part of the face and place system. If we apply Searchlight multivariate pattern classification, this is using the Searchlight toolbox that Francisco Pereira programmed. What you can see is, again, it looks pretty consistent with the standard view. So we have bilateral FFA on the right and the homologous region on the left, We also have a little bit of information down here, which might be similar to this area. It's a little bit more anterior, and a little bit of right-sided occipito-temporal region that can be useful for decoding faces from other objects in this data set. If we make the searchlight bigger, the signal seems a lot more distributed, but it's still roughly in posterior temporal occipital areas, as the canonical view would suggest. So this all looks consistent with the canonical view. If we decode the whole brain using L1 regularization, we also get a result that looks pretty consistent with canonical results. So what I'm showing here with the lasso is all the voxels that reliably received a non-zero coefficient relative to permutation testing, and where the color is showing the proportion of subjects in which that coefficient was positive, okay? So red areas had a positive coefficient, suggesting that elevated bold activity helped to predict faces. And blue areas have negative coefficients, suggesting that elevated activity helped to predict not a face, right? So here you can see this nice gradient between face, non-face comes out in the coefficients from the L1 whole brain decoder. And again, we get this right-sided occipito-temporal patch where elevated activity seems to signal faces according to the lasso solution. So all of these approaches from the literature, one might look at and say, yeah, we've confirmed the kind of modular view of face representation in the brain. But if we apply the sparse overlapping sets lasso, we get a very different picture, a much more widely distributed view of where the information is. So again, this is the same style of plot as lasso. The color indicates the proportion of subjects who receive a positive beta coefficient in their decoding model. Blue is indicated negative coefficient. Green are areas that received non-zero coefficients more often than you'd expect from permutation testing, but where the direction of the coefficient was variable across subjects, and so didn't consistently lead to the same direction. So we look at this kind of highly distributed result, and you have to ask, is this junk? This is some new decoding technique. Is this a meaningful result? So there are three follow-up analyses I wanted to tell you about. The first is, if we look at these coefficients, decoding, is it a face or not? And look at the places where the coefficients all tend to be positive across the subjects. What we can see is that this is basically picking out the social cognition network. We have bilateral anterior temporal lobes, which we know are important for representing individuals. We have the right orbitofrontal cortex, and we have this sort of anterior superior medial prefrontal cortex, all areas that are thought to be involved in representing our knowledge about others. And the places where the coefficients are negative, so that high activity signals it's not a face, we've got basically the dorsal visual stream, and we've got the perihippocampal place area, and we have this sort of left premotor area, all areas that seem to be involved in representing artifactual objects and object-guided action. So in other words, non-social kinds of things. So at least the kind of layout of the coefficients seems to be consistent with a lot of other literature outside of the face processing world. Secondly, it's not the case that you always get this very widely distributed signal, no matter whenever you apply SOS lasso. So on the same data set, if we train the model to decode, is it a place or a non-place, what we see is basically a replication of the classic perihippocampal place area result. The regions that tend to be more signal that the stimulus is a scene versus a non-scene are nicely localized to the perihippocampal place area. So this is sort of a proof of concept that SOS lasso can discover localized signal where that signal exists. And finally, there's some concern that all of this distributed stuff is just a spurious result that is somehow allowing the decoder to better exploit signal that might be really localized within the face and place system. For instance, if there's whole brain correlated noise, you can improve the performance of a classifier by placing coefficients on extra system areas that serve to subtract out the noise that is distorting the signal carrying areas. What we did was to fit stimulus decoders to voxels either across the entire cortex or only within Canwisher's ROIs, plumped out by about 7 millimeters so that we're getting everything, or only to voxels that are outside of the Canwisher ROIs. And then we can look and see how well do the decoders do. If all the signal is really in the ROIs, then when you fit a model outside the ROIs, you should do worse at decoding. So these are means in individual subject decoding holdout cross-validation accuracy for decoding faces versus places in the whole brain. Here's what we get if we decode only from the Canwisher ROIs. These are both face and place areas plumped out by 7 millimeters. If we decode outside those areas, this is what we see for holdout cross-validation accuracy. If anything, we're doing slightly better decoding outside of those systems than we are inside the systems. So what does that all mean? For me, those 3 points suggest that the SOS Lasso result is not some weird artifact, it's not spurious. We know for sure that the widely distributed signal that SOS Lasso finds isn't found even with the very large searchlight that we used, right? So that tells us that the information it's exploiting has to be non-local. If it was just within some big patch of cortex, Searchlight would find it just fine. It doesn't. It must be the case that distal regions are cooperating in some way to represent that information. And the signal looks a lot more distributed as long as you're applying a method that is capable of finding that distributed signal. In the simulations, we showed that SOS Lasso is the only method that can find anatomically dispersed distributed signal. When we apply that approach to the real brain data, it looks like face representations are engaging whole networks, not just local modules. Moreover, that's not guaranteed result, right? When we decode scenes, we get a localized representation. And so it becomes an empirical question of all the results in the literature that are claiming some system is functionally or anatomically localized based on brain imaging data or other data. It now becomes an open question. If you were to look at that same data using structured sparsity, would the representation seem to be more distributed? And if so, that suggests a very different account of how information gets represented in neural activity. In this particular domain, I think the data show that face perception is generating a quite widely distributed neural signature across much of the brain, not engaging a small set of modules in posterior temporal occipital cortex, as is the received view. More generally, I think that neural network models can help to explain why we get different patterns of results across methods. I've shown that with a super simple model. I think if you followed along, you can understand why the different approaches are leading to somewhat different pictures about where information is in the brain. And it so happens there are tons of models from the 80s and 90s even that we know are good at simulating human behavior that can maybe be brought up to speed with contemporary tools and exploited for this purpose, for helping us to understand different patterns of functional brain imaging. Clearly, I did not allot enough time to go through the second part of the talk, which has to do with decoding ECOG signals and neural dynamics. But hopefully I was clear about the material on functional brain imaging, and I'm very happy to entertain any questions that you guys might have right now.
Thank you, Doctor. We now move to the QA part. If anyone has any questions, please either add them to the Q&A part or raise your hand so that we can give you voice permissions. We don't have your voice. Now you have your voice.
Can you hear me?
Yes.
Thank you very much for your interesting presentation.
Yeah.
And is there any question? So I have one question about the modeling of noise in your model.
Yeah.
Actually, I saw that you have added some Gaussian noise to model. Is it true?
Yes, that's true. So we don't, so in that little model, we don't have noise intrinsic to the activations, but we imagine that when you're measuring the neural response, that there's measurement error that we model as Gaussian noise.
Okay. So there is some evidence that the noise in our brain is not Gaussian, and some other alternatives for the modeling is Levy flights or alpha stable distribution. What's your opinion about modeling the alpha stable noise to this model?
Yeah, so you're asking whether the properties of the noise matter for the decoding?
Yeah. Yes.
Yeah. So I suspect that if we had better models of noise in the decoder, we could do a better job at pulling the information out, for sure. What I'm less sure about is, you know, in all of the simulation, but also the real data where it matters, I think, you know, we're just using cross-validation to assess model performance. And at least in the fMRI case with these data, the slides I have here are a little bit out of date, but with better parameter search tools, we've gotten these decoding accuracies really up to about 95% correct, which suggests to me that we're at kind of at the resolution where having the right model of the noise doesn't really matter. But it will, I think it will matter as we get to less easy kinds of decoding problems.
Okay. Okay. So there's no matter that using any simple models of noise.
Yeah, I mean, I'll just elaborate a little bit, which is that one of the big challenges for whole brain decoding of this kind is, I sort of rushed through this bit, but you know, if noise is distorting different brain systems and the source of the noise is, you know, causing the noise components to be correlated across these features, right? Then, you know, if one voxel carries signal and it's distorted by that noise, and the second voxel carries no signal but is distorted by the same noise, then you can improve your decoding by placing opposite weights on those two features. Place a positive weight on the signal carrying feature and a negative weight on the non-signal carrying feature. Then the non-signal carrying feature subtracts out the noise from the signal carrying feature and boosts the performance of the classifier. And so now when you look at interpreting the weights, you don't want to, you know, the second feature might attract a weight because it's only boosting the signal in the real system, not because it's self-carrying signal. And that's actually, I think, a very thorny issue for these kinds of approaches. The best that we've been able to do in this kind of case is to you know, show that when you lesion out, so if you remove all of the face and place system, you can still decode from what's left over very accurately. right? So we know for sure that there has to be real information outside of those canonical systems, but we still don't know, of all of those voxels that the method selects, which ones are just subtracting out signal correlated noise and which ones aren't. So any insight or input on that, please email me.
Okay.
So there is also a question in the Q&A part. You can see it in the QA part of the Zoom application. And if you prefer, I can read it to you.
I can't. Maybe if I stop my sharing, I'll be able to see. Oh, it's in the chat?
Yes, it's QA day. Yeah.
QA and A part.
Oh, there it is. I see. Yeah, so the question is, what kind of signal to noise ratio do you need to combat the noise? This is another thing that's sort of interesting. The answer really depends on the kind of model regularization that you use. So if you regularize with the L2 norm, which is just the square of the coefficients, you know, putting a big coefficient on a single feature incurs a huge cost, right? The bigger, if you put a coefficient of 10 on a single feature, it incurs a cost of 100. But if you have 10 correlated features and you put a coefficient of 1 on each of them, right? The net effect is still the same. You're getting 10 because they're all going together, but the total cost on that is now only 10 instead of 100. So the L2 norm likes to spread coefficients across many features. And so what that means is that if the noise is independent on each feature, spreading across all those guys, the noise will cancel out and you can have a much lower signal to noise ratio at any individual feature, right? Because you're essentially averaging across a bunch of correlated stuff. Compared to if you look at the L1 norm, it's going to much prefer to put as much weight as it can on an individual feature. And so if the signal to noise ratio at an individual feature is very low, it's going to have a hard time finding that signal at all. And so I think there's, you know, taking L1 and L2 as two kind of very common regularization approaches, there's probably some clever things to be done with regard to grouping coefficients so that you're kind of grouping things at a level that takes the signal to noise into account so that that averaging is going to boost signal without including a lot of random features.
So is there another question?
Well, seems not.
Okay.
So I believe we can conclude today's talk. Thank you, Doctor, for joining us today. It was a really interesting topic, and I believe that's it.
Yeah, my pleasure. Thank you guys for inviting me, and good luck with the rest of your conference.
Thank you very much.
Thank you very much.
Goodbye.
Thank you.
Bye.
Goodbye.